Hey guys, welcome to the fifth part of a five-part lecture series over the endocrine system. Today we're going to be talking about the endocrine regulation of blood glucose or blood sugar, and we're going to draw a focal point to diabetes, specifically diabetes mellitus. So as always, I've provided you with a set of learning objectives: live them, love them, learn them, and I've also provided you with a lecture handout. I'd have that lecture handout out and available, and I would track through the different learning objectives as we go through them so you know what the important points of each slide or each objective that we're going over is. So, learning objective one is just distinguishing between an exocrine and an endocrine gland. Endocrine glands are glands that secrete chemical messengers directly into the bloodstream, and those chemical messengers are swept through the bloodstream until they reach a distant target cell where they bind to a receptor and stimulate a response. Any chemical messenger secreted directly into the bloodstream is by definition a hormone. The endocrine function that we're going to be focusing on today is the endocrine function of the pancreas. So we all know what the pancreas looks like macroscopically. We can identify it. It's directly posterior to the stomach. We've probably had a cat dissection at this point, and we've definitely looked at the models. Now, histologically, the pancreas can be broken into two types of tissue. The first type of tissue is what's called pancreatic acini or pancreatic acinar cells. This is the exocrine pancreas. So this dark staining tissue out here, the job of this dark staining tissue out here is to produce pancreatic enzymes and pancreatic juice that enter into the pancreatic duct, travel to the lumen of the small intestines, and are released there where they facilitate the breakdown of macromolecules in our food. Things like proteins into amino acids or, polysaccharides into monosaccharides, whatever it is. So that's an exocrine function, and the exocrine pancreas plays a really important role in digestion. The endocrine pancreas, you get little pockets or islands of endocrine tissue referred to as islets of Langerhans or pancreatic islets. They stain much lighter than the surrounding exocrine tissue, and they're really easy to see or observe when you look at them under a histological image. So if I said identify the structure indicated by the pointer, that would be a pancreatic islet or an islet of Langerhans. Islets of Langerhans consist of two types of cells that you're going to need to be familiar with in this class. Kind of toward the outside, and I would never ask about a cell type on an image like this, but toward the outside you have what are called alpha pancreatic cells. Alpha pancreatic cells produce a hormone called glucagon. They do that in response to low glucose levels. So glucagon raises blood glucose. Clustered kind of toward the center are what are called beta pancreatic cells. Beta pancreatic cells are responsible for producing a hormone called insulin. And what insulin does is it lowers blood glucose levels. So when you're thinking about the first follow along, kind of look at that image and make sure that you can identify a pancreatic islet and that you know we're looking at the pancreas. Now, when we think about alpha and beta pancreatic cells, let's say blood glucose levels are low, so those little dots represent glucose. When blood glucose levels drop, the pancreas detects that. Specifically, alpha pancreatic cells function as receptors. They detect that information. They function as control centers. They process that information, and they release an effector, which is a hormone called glucagon. Glucagon then works on the liver or has target cells in the liver where it has the exact opposite effect of insulin. It triggers glycogenolysis, which is the breakdown of glycogen into glucose. That glucose is released into the blood. Gluconeogenesis, which is forming glucose from non-glucose precursors. It's making new glucose, releasing it into the blood. The collective effect of that is an increase in blood glucose, hopefully, right, to reestablish normal reference range limits. On the flip token, when blood glucose levels are very high, beta pancreatic cells function as receptors and control centers, and they release an effector, which is the hormone insulin. Insulin then travels to like skeletal muscle fibers, adipose cells, where it binds to receptors, extracellular receptors because it's a protein hormone, and it triggers the insertion of a little transporter called a GLUT transporter. When the GLUT transporter is inserted into the surface of the membrane, these cells can suck up glucose. When glucose moves from the blood into the cells, blood glucose levels decrease and homeostasis is reestablished. So when we're looking at this guy right here, Know that the pancreas, when you look at it histologically, is broken into those exocrine and endocrine portions. Exocrine pancreas produces digestive enzymes that enter into the pancreatic duct and ultimately empty into the small intestines, where they're responsible for breaking down macromolecules. The endocrine pancreas, on the other hand, is responsible for regulating blood glucose, and within those islets of Langerhans you find alpha and beta pancreatic cells. Now, when we think about alpha and beta pancreatic cells, remember I told you alpha cells kind of clustered toward the outside and beta pancreatic cells kind of clustered toward the inside? We kind of know what we're dealing with now, right? So we have this islet of Langerhans, and the alpha pancreatic cells produce glucagon, the beta pancreatic cells produce insulin. Now, when we think about the discovery of insulin, right, we didn't know in the early 20th century what the pancreas really did. We understood that it played a role in digestion, but diabetes, type 1 diabetes mellitus, at that time in the early 19th century was a death sentence, was an absolute death sentence, right? Kids would waste away, essentially starve to death, urinate themselves to death, and die of diabetic ketoacidosis or drops in blood pressure, right? Whatever it was a death sentence. Until a guy named Dr. Banting came along, and what he did is he said, I wonder if the pancreas has something to do with this. So he took dogs, he removed their pancreas, he essentially induced a diabetic state and went, okay, well, after you remove the pancreas, the dog develops diabetes. The pancreas obviously has something to do with the regulation of blood glucose. Diabetes used to be called honey urine disease, by the way. And the way that you diagnose it is you just dip your finger in and take a taste, because there's so much glucose in the urine, it actually has a sweet taste to it. So, dog developed diabetes, right? Then he essentially took the pancreas, put it into a blender, went brrr, sucked out the juice, injected it back into the dog, and he could find that he could treat the diabetic state, the state of elevated glucose levels. And that was kind of a magical moment that led to the discovery of insulin. The discovery of insulin has saved millions of lives. Tremendous, tremendous contribution to medicine. Now, when we think about insulin, insulin's responsible for regulating the concentration of blood glucose. And we know that glucose is a really useful molecule because it's easily burned for energy. So it's kind of an immediate source of energy that can be used or converted to ATP, which the cells can readily use. When glucose enters the body, it has one of three fates. One of three things is going to happen to glucose. Initially, if cells need energy, the moment that glucose enters into a cell, it'll undergo what's called cellular respiration. So we burn glucose at the level of the cell in the presence of oxygen in order to make ATP. The byproducts of that are carbon dioxide and water. This is called cellular respiration. That's the burning of glucose in the presence of oxygen in order to make ATP with CO2 and water as the byproducts. In fact, if you wonder why you breathe, why you inhale in order to get oxygen to fuel this reaction, and you exhale in order to get rid of the byproduct carbon dioxide, right? That's where it's coming from. It's coming from cellular respiration. Now, if the immediate energy reserves or the immediate energy needs of the cell are met, cells can carry out what's called glycogenesis. This is where you convert glucose to glycogen. And remember, glycogen is just a polysaccharide that consists of multiple glucose molecules linked together covalently. It's a storage form of glucose. If you have plenty of ATP, why continue to burn glucose? Why not store it for a rainy day? Where we store most glycogen in our body, it's the storage form of glucose in animals, is in the liver and in skeletal muscle. Right? Finally, if our glycogen reserves are filled, glucose can be converted to lipids, right? We can use glucose in a series of enzymatic pathways to make lipids. So we used to think that fats were responsible for obesity. We now know that sugars are actually a much more dangerous culprit. Because once you have those energy stores readily available, right, fat storage begins, and glucose can be used to make fat or be used in lipid synthesis. So when glucose enters the body, that's kind of chronologically what happens to it. Now, when we think about sources of blood glucose, we're going to be talking about fed and fasted states. A fed state is after you eat. For about 8 hours after you eat, you're considered in a fed state. So when you're looking, ingested glucose, and you'll see here it drops for about 8 hours. After 8 hours of not having a drop of glucose either in food or in drink, you are considered to be in a fasted state. So we're going to be talking about fed and fasted states and what the predominant hormone is in each one of those states. So when we think about this, hormones often have antagonistic controls. If you need to reduce the concentrations or levels of blood glucose with the use of insulin, you also need a hormone that's going to increase the concentration of blood glucose. By the way, when you're looking at your graph on your handout, pay attention to what the graph is actually telling you. Are you looking at insulin concentrations, or are you looking at glucose concentrations? And what do those fluctuations in glucose concentrations tell you about fed versus fasted states? Because in fed states, when blood glucose levels are high, the dominant hormone is going to be insulin. And insulin, when it binds to the receptors in like skeletal muscle fibers and adipose tissue, upregulates a number of different processes. One, it triggers the insertion of transporters that can physically move glucose into the cell, and that's the primary mechanism by which it lowers blood glucose. Then it triggers the burning of glucose, which is called glucose oxidation. So that's cellular respiration, glucose oxidation, cellular respiration, same thing. We get glycogen synthesis once we have our immediate energy needs met. And then we get fat synthesis. So you think about that and you go, well, insulin can actually, if you have excess glucose and insulin's always being released, can actually contribute to things like obesity. It makes adipose cells little fat storage factories. So insulin is the dominant hormone in fed states. Glucagon, on the other hand, is the dominant hormone in fasted states. So if you've gone over 8 hours without touching anything that has any glucose in it, right, you're considered to be in a fasted state. And when you take certain clinical exams, like certain blood diagnostics, you have to be in a fasted state. It's one of the things you should always ask your doctor if they order you some labs. Say, do I need to be, it doesn't matter if I'm fed or fasted. So in the fasted state, glucagon does the exact opposite of insulin, right? If you're fasting, blood glucose levels are low, so glycogenolysis is just the breakdown of glycogen to release those glucose molecules so they can be used by the body. Gluconeogenesis is the formation of glucose from non-glucose precursors to make new glucose molecules that can be released into the blood. And ketogenesis, because the body still needs energy, one of the ways it can get energy is by the breakdown of fat. But when you break down fat, you release these very acidic byproducts called ketone bodies, right? So rather than building up glycogen and fat, it breaks down glycogen and fat and it makes new glucose. That glucose is released into the system and blood glucose levels go up, because if you're fasted, you're going to want to bring your glucose levels up so you have enough energy reserve to do work. Now, when you think about the stimuli for the release of insulin, we all know that elevated blood glucose concentrations can trigger the release of insulin. And that's, by a long shot, the most important stimulus for the release of insulin. That's the humoral stimulus. But there are also two other stimuli that factor into insulin release and therefore how our body handles blood glucose. And it's really important to kind of mention those here. And if you're looking at the Learning Objective 5, I want you to really pay attention to what I'm asking with those graphs. What are we actually asking there, and why is that the case? So when you eat a meal that contains glucose, when you swallow and this stuff moves into your GI tract, you get distension of the GI tract wall. That's detected by stretch receptors. That information is taken to the central nervous system, where a parasympathetic output along the vagus nerve, the pre- and postganglionic neurons of the vagus nerve, is stimulated. The vagus nerve feeds into all of the major thoracic and abdominal viscera, meaning thoracic and abdominal organs. And essentially, this parasympathetic output, this direct parasympathetic output, right, you have a neuron telling these beta pancreatic cells to release insulin, right? It's kind of a preparation. So you go, okay, that's an immediate release of insulin because you want to handle that glucose right when it enters the system. Another, right, so that would be neural. Another stimulus for the release of insulin is when carbohydrates are present in the GI tract. There are little cells that line the small intestines called enteroendocrine cells, and they release two hormones, glucagon-like peptide 1 and gastric inhibitory peptide. Now, these are hormones with multiple targets. One of their targets is beta pancreatic cells, and what they trigger in beta pancreatic cells is the release of insulin. That's your hormonal stimulus. But the most important stimulus by a long shot is your humoral stimulus. So once you get the absorption of glucose, plasma glucose concentrations are going to increase. So we have an increase in the concentration of a factor in the blood, and that's what triggers this endocrine pathway. That's why it's called a humoral stimulus, because the humors are the fluids of the body. So increase in blood glucose levels will trigger these beta pancreatic cells directly. So will amino acids, which is why drinking proteins, or like protein shakes, can actually quite good for insulin sensitivity. But that's another thing altogether. This is where you're going to get the whopping percentage of the release of insulin, right? Is this guy right here, these plasma glucose concentrations. But all three of these are really important. And why do you think on your diagram that you have, you see that spike? Because you're measuring insulin, not glucose. And you're measuring insulin spikes in response to oral glucose versus IV glucose, because it's really important to understand how the body's handling that. Why do you get that gigantic spike of insulin, right? Why do you get that gigantic spike of insulin? It's called the incretin effect. Think about it from this perspective. What are the stimuli for the release of insulin, and why do you get a different insulin response if you drink glucose as opposed to if you run an IV of glucose, which is really important from a clinical perspective. Now, learning objective 6 is the mechanism of beta cell sensing. So when blood glucose levels are elevated and you have the humoral stimulus, there's a transporter in the surface of beta pancreatic cells, a protein transporter called a GLUT2 transporter. This is what's known as an insulin independent GLUT, meaning GLUT2 transporters are going to be embedded in the membrane of beta pancreatic cells, whether insulin is around or not. And that's a good thing because these cells need to be able to transport glucose independently of insulin. So we call GLUT2 transporters insulin independent GLUTs. So glucose moves through this GLUT2 transporter, right, which is a protein, meaning that DNA to RNA, RNA to protein, protein function. Our genes provide information for this. And in the cell, that glucose is used to make ATP in the process of cellular respiration. That ATP then comes and closes potassium channels. When potassium channels close, potassium can no longer leak out of the cell, and because potassium has a positive charge, it's going to cause the cell to depolarize. So one of the things the electrochemical gradient is used for, right, is in endocrine sensing. It's not just action potentials. The cell depolarizes. When the cell depolarizes, voltage-gated calcium channels are activated. Calcium rushes into the cell. Calcium is always necessary for exocytosis. So that calcium mobilizes these vesicles and triggers the exocytosis of insulin. When you're looking through your handout, think about the way the mechanism of action of sulfonylurea drugs. Think about what they actually do. Now, when we think about the secretion of insulin, one of the things that I want to highlight is there's a processing pathway for any protein that's being shipped out of the cell. So insulin is made in the rough endoplasmic reticulum. It's called pre-proinsulin. Pre-proinsulin consists of 3 chains. We have a beta chain, a C-peptide chain, and an alpha chain, right? Pre-proinsulin has a little marker on it called a signal sequence that actually tells the Golgi apparatus where that particular protein is going to go. In the Golgi apparatus, pre-proinsulin, the signal sequence is cleaved off and it turns into proinsulin. Proinsulin is then shipped, right? And on its way out, proinsulin is cleaved to form insulin and C-peptide. then insulin and C-peptide are released in a 1-to-1 ratio. Now, insulin is broken down very quickly by the liver. So as insulin travels through the body, the liver breaks it down really quickly, which makes it really problematic to try and measure insulin directly. So because C-peptide is released in a 1-to-1 ratio with insulin, a lot of times clinicians will order a C-peptide test. When you see a C-peptide test ordered, that's really a measure of insulin. It's an indirect measure, but because these are released in a 1-to-1 ratio, C-peptide is really useful because it doesn't break down very quickly, meaning that it remains elevated in the blood for a prolonged period of time. It doesn't get processed and broken down by the liver quite as quickly. It sticks around for a lot longer, making it a lot more stable way to measure how big of an insulin spike you had. So C-peptide is really kind of an indirect measure of insulin. Now, when you think about learning objective 7, you heard me talk about insulin-dependent and insulin-independent GLUT transporters. How does an insulin actually exert its effect? What's the mechanism? And remember, as physiologists, we always care about mechanism. So insulin is a protein hormone. It's released by beta pancreatic cells, and it has two primary targets that I want you to know. Skeletal muscle, right? So skeletal muscle tissue and adipose tissue. When insulin binds to protein receptors in skeletal muscle or adipose tissue, it triggers a signaling pathway in which the primary response is to insert a transporter into the membrane called a GLUT4 transporter. Now, all these secondary pathways down the road, changes metabolism, that's where you get increased, so we're talking about lowering glucose, that's where you get increased glucose burning, the synthesis of glycogen, the synthesis of fat, right? That's those changes in metabolism. But the primary thing that insulin does is it triggers the insertion of a transport protein to be put into the cell membrane. And that transport protein you find in skeletal muscle fibers and adipose tissue is called a GLUT4 transporter. These transporters can then move glucose from the extracellular fluid, which is like the blood plasma and the interstitial fluid, into the cell so the cell can use that glucose. Without these GLUT transporters, right, you can't move glucose into the cell, so the glucose just stays outside of the cell and builds up and builds up. You need these to lower blood glucose levels. It's the only way to get glucose into the cell. When we say a transporter is insulin-dependent GLUT, what we're saying is insulin has to be bound to its receptor, triggering this pathway in order for GLUT4 to be found in the membrane of skeletal muscle fibers and adipose cells, right? If no insulin is around, this GLUT4 transporter will not be found in the membrane, and glucose has no way to get into the cell, so it remains outside the cell in places like the blood. Now, when you think about diabetes mellitus, it's not a single disease entity, but it's a group of metabolic disorders characterized by hyperglycemia, meaning blood glucose levels that are higher than reference range or higher than normal. It is a massive problem throughout the developed world. Every year, 800,000 people develop diabetes, and about 54,000 people die from that illness. If you compare that to COVID, diabetes mellitus actually takes a much, more pronounced toll on the world's population. So when you think about diabetes is not a single system disorder. A lot of people go, well, it's just, high blood glucose. Well, one of the problems with glucose is if you've ever spilled Coca-Cola on your hands, you'll realize that glucose is sticky. Now, that sticky property of glucose, that ability to kind of adhere or stick to things, is really problematic. And most of the consequences of diabetes arise from the fact that glucose is just sticky and it sticks to everything and it gums things up. So when you think about the leading cause of kidney failure is complications related to diabetes. Why? Well, in those filtration membranes in the kidneys, glucose essentially sticks to them, gums them up, and prevents the kidneys from being able to filter your blood plasma anymore. That's massively problematic because then toxins build up and you die. So this person's on dialysis here. Leading cause of blindness, also diabetes, right? Blood glucose sticks to the capillary exchange networks and, for example, the retinal capillary networks in the retina of the eye. And when glucose sticks, it makes it very difficult to exchange oxygen and nutrients. So the retina of the eye starts to literally die because there's so much glucose, making it so difficult to exchange at those capillary networks. Same thing, having elevated glucose lowers your blood pressure and makes it difficult to exchange at capillary networks, which really becomes problematic with things like appendages, which need good capillary perfusion. So the leading cause of amputation. My point being is it's not just high blood sugar. High blood sugar impacts everything in your body. So when we think about classifications of diabetes, we're going to focus on type 1 and type 2 today, but just so you know, there's an entire slew of different classifications of diabetes mellitus, all of which share one commonality, and that is people have elevated blood glucose levels. The specific cause differs from different types. Our focus today is going to be type 1 and type 2, but just know there's a bunch of other types of diabetes. So type 1 diabetes, and this is where we get into learning objective 8, right? Type 1 diabetes is an autoimmune disease that destroys beta pancreatic cells for an unknown reason, right? We don't really get it. We're starting to try kind of figure those things out, but we don't understand why the immune system just turns on those beta pancreatic cells. It tends to onset in childhood. It can onset later in life, but it tends to onset in childhood, and it is a disease of hyposecretion, meaning that if beta pancreatic cells get destroyed, you're not going to be able to release insulin into the blood. If you can't release insulin into the blood glucose levels are going to be elevated, and they're going to cause all the problems that elevated blood glucose does. It's going to stick to and gum up everything. So type 1 diabetes is actually a little bit easier to treat in some respects because you can, if somebody needs insulin or is insulin dependent, you can give them insulin, right? You can charge out the wazoo for it, but you can give them insulin, and if people are regimented and good about it or get an insulin pump, it's a manageable disease. Now, it used to be a death sentence. Type 2 diabetes, on the other hand, is not a disease of hyposecretion, it's a disease of insensitivity. So we often refer to type 2 diabetes as being a disease of lifestyle. When you're constantly spiking blood glucose, insulin, right, is always going to be elevated. And if insulin's constantly elevated, those cells are going to start, the insulin receptors are either going to stop interacting with insulin or the cells are going to downregulate and remove those insulin receptors. So in type 2 diabetes, beta pancreatic cells remain functional. You have plenty of insulin, at least earlier in the disease state, right? The problem is that insulin isn't working anymore. This is considered a disease of insensitivity. So we have insulin dependent and insulin independent. I don't really like those terms, right? Type 1 and type 2 diabetes. It's not a disease of hyposecretion. It's a disease of insensitivity. Insulin stops working like it once did. So when you think about it, this image isn't completely accurate, but I think it does a pretty good job. In type 1 diabetes, you have an autoimmune disease that destroys beta pancreatic cells, so you can't produce insulin. So those insulin receptors are there, there's just no insulin signal. So if you can introduce insulin and be regimented about when you eat and when you introduce the insulin, right, you can manage the disease pretty effectively. Type 2 diabetes, on the other hand, you have plenty of insulin. It's just these insulin receptors have become insensitive because they're constantly bombarded with it. It's like wearing out a lock with a key by just putting it in and out, right? Going, You wear out the mechanism. Well, these guys get worn out and they can no longer elicit a response. This tends to onset a little bit later in life. We used to call it adult onset diabetes, but uh, it's becoming very common. We no longer call it that anymore. It's becoming very common for children as young as like 5 years old to develop type 2 diabetes just as a consequence of what's going on with lifestyle factors and, genetic predispositions. Um, so, God, if people would follow lifestyle suggestions, everybody says, well, why don't we practice preventative medicine? Want to know one of the biggest barriers to preventative medicine? It's patient adherence. You tell somebody to stop doing something, it's unlikely that they're going to stop doing something. And I get it. I don't even fault people for that, but patient adherence is a big problem, right? If you tell me, hey, stop doing this, I'm going to be hard pressed to do that, right? Especially if it's a vice of mine, and I have plenty. So, when you think about type 2 diabetes, it's often associated with what's called metabolic syndrome. And metabolic syndrome is characterized by things like central obesity, high blood pressure, high triglyceride levels, and the fact that insulin isn't working very well anymore. All of these things kind of put together can dramatically impact the health of the entire organism and lead to really kind of prolonged, painful, chronic states. So when metabolic syndrome kicks in, you're kind of late in the disease game. Now, can you turn that around? Yeah. The best way to do that is not pharmaceutical, although there are some good pharmaceutical alternatives. It's lifestyle changes, which we're going to talk about. So when you think about a thought question like this, right? We're going through it as a class. I just want to highlight what you're looking at. These are destroyed beta pancreatic cells, and what you're looking at there is the accumulation of scar tissue. So think about the question you've been given, and then we're going to talk about the reason that some of those symptoms present the way they do. But bottom line here is the classical symptomology is polyphagia, eating too much, polydipsia, urinating too much, and polydipsia, drinking too much, and polyuria, which is urinating too much. So now we're on learning objective 9, right? And you've done that little case study. And we're going to distinguish between insulin-dependent and insulin-independent GLUTS. So insulin-dependent GLUTS, GLUT1, found in red blood cells. That's how red blood cells suck up glucose. Because glucose is sticky, when you think about a hemoglobin A1C, hemoglobin A1C is just measuring essentially the weight of hemoglobin. Because glucose sticks to hemoglobin, the more glucose you have, right, the more glucose is going to stick to hemoglobin, the heavier that hemoglobin is going to become. So if it's above 5, right, and there's a metric there, it means that you have heavy hemoglobin because a bunch of glucose is stuck to it. A hemoglobin A1C is a much better measure of long-term glucose regulation because a finger stick is just a snapshot. Red blood cells live about 3 months, so a hemoglobin A1C can give you an idea about like 3 months of what glucose regulation has been like. And if glucose is sticking to that hemoglobin's going to get heavy. That's what a hemoglobin A1C actually measures, is the essentially weight of hemoglobin, right? And the heavier it is, the more glucose is stuck to it, which is problematic. So GLUT1 transporters are insulin dependent, meaning they're not in the surface of a red blood cell unless insulin has bound to its receptor and trigger that pathway. GLUT4 transporters, which are found in resting skeletal muscle tissue and adipose tissue, are also insulin dependent. GLUT3, it's all right. GLUT2, on the other hand, you find GLUT3 in neurons, and there's been a lot of controversy over that one. GLUT2 transporters. GLUT2 transporters are what we refer to as insulin independent. So you have beta pancreatic cells in the liver. Beta pancreatic cells, you don't want to have, they're what release insulin. You don't want their sensing mechanism to be disrupted, so that's good. And the liver, right, both sucks in and releases glucose. It plays a really important role in glucose metabolism, and you don't want it to be, its ability to move glucose to be dependent on the presence of insulin. So when you think about a fasted state, right? In a fasted state, you have low levels of glucose, and that insulin receptor doesn't have any insulin on it because glucagon would be the predominant hormone. Now, in a fed state, glucose levels spike, insulin, right, binds to its receptor. The primary impact it has, the way that insulin lowers blood glucose, is it binds to the receptor in either skeletal muscle tissue or adipose tissue, and it leads to a pathway in which these GLUT transporters that are in the cell, right, attached to these little vesicles, are actually mobilized and inserted into the cell membrane. Once these GLUT transporters are inserted into the cell membrane, the cell can suck up glucose. And remember, GLUT4 transporters are insulin dependent, right? So, but in the absence of insulin, you're not going to get any of those GLUT4 transporters in the cell membrane because they're going to be inside the cell in these little vesicles waiting for the signal to release. In other words, if you don't have insulin or insulin's not working, you can't actually move glucose into the cell. And if you can't move glucose into the cell, the cell can't use glucose, so it has to use other things, right? So that's what insulin dependence means, and that's the most important mechanism. So if I ask you if insulin spikes in a healthy individual, right, which of the following would be true? Definitely have on your radar that when insulin binds, its primary effect is to insert these GLUT4 transporters into the membrane of skeletal muscle cells and adipose cells that can actually suck glucose out of the system and lower blood glucose levels by moving glucose into the cell. Once glucose enters the cell, it's either burned for energy, stored as glycogen, or used to synthesize fat. Insulin independent, for example, in beta pancreatic and liver cells, doesn't really matter if insulin is present or not. Those GLUT2 transporters are always going to be in the surface of the cell membrane. One of the important things to realize is that GLUT transporters will move glucose along a concentration gradient. So if glucose levels are high in the cell, right, glucose can move from inside the cell to outside the cell, or if they're high outside the cell, glucose can move from the outside in. That's important, this bidirectional movement, especially in the liver. So the liver has insulin receptors, right? But it's just that these insulin receptors don't actually lead to a pathway that stimulates the insertion of these transporters. These GLUT2 transporters are what are called insulin independent, meaning they're always going to be in the membrane of the cells that they're found in. For this class, know that you find GLUT2 transporters in beta pancreatic cells and in liver cells. In the liver, it's really important because the liver needs to be able to move glucose in multiple directions. So, for example, when glucose levels are low and glucagon kicks in, glucagon's primary target is the liver. It triggers the breakdown of glycogen, right, into these individual glucose molecules that need to be able to get out into the extracellular fluid, right? So, yeah, and on the flip token, when glucose is elevated, we need to be able to store it as glycogen and use it to make fat. So there's a two-way transport that's taking place there. Now, one of the exceptions to this GLUT4 in skeletal muscle rule is, notice that I've said resting skeletal muscle. When skeletal muscle starts to exercise, and exercise doesn't have to be doing P90X, exercise could just be walking or jogging, right? Exercise could be anything. It could be mowing the lawn. But when skeletal muscle fibers activate, it actually leads to a mechanism that overrides the necessity for insulin to be present, and these GLUT4 transporters can actually be inserted into the surface of the cell's membrane where they can transport glucose into the cell. So with type 2 diabetes, there are a lot of different medications like sulfonylureas and metformin and things like that, but one of the best things you can do, one of the best interventions that will slow the disease process down is changes in diet and lifestyle. And those lifestyle changes are just get more exercise, because when you exercise, it inserts those GLUT4 transporters, override that insulin dependence, and are able to suck glucose into those skeletal muscle fibers. It's a protective mechanism to make sure that skeletal muscle gets plenty of glucose if it's working, right? Especially if you're doing something like running away from something. So exercise is really, really important. Now, when you think about learning objective 11, hyperglycemia is defined as elevated blood glucose. If you're fasted, meaning you haven't had anything with glucose for 8 to 12 hours, anything 125 milligrams per deciliter and above, you're considered overtly hypoglycemic. 70 milligrams per deciliter and below is considered hypoglycemic. So some people don't suffer hyperglycemia, some people suffer hypoglycemia, and normoglycemia is between 70 and 100 milligrams per deciliter. Now, when we think about diabetes, if you're between 70 and 100 milligrams per deciliter on a fasting glucose test, you don't have diabetes. If you're between 100 and 125 milligrams per deciliter, you are pre-diabetic. That is the moment when you should start taking very seriously your health, because you do not want to develop overt diabetes, right? Losing just 7% of excess body weight can do wonders for your metabolism. It reduces the chance of developing type 2 diabetes tremendously. And then if you are above 125 milligrams per deciliter, you are overtly diabetic. So this would be fasting blood glucose. Now, one of the things we're doing in the lab next week is what's called a glucose tolerance test. And in a glucose tolerance test, what we do is we actually have a patient come in fasting and we give them a sugary drink, a glucose drink that they have to chug within 5 minutes. Then we take blood glucose levels every 15 minutes for 2 hours. And after that 2 hours, here we have the diagnostic criteria for where blood glucose should be. So after 2 hours, no matter how much glucose you ingested, if you ate a bloody birthday cake, it should be below 140 milligrams per deciliter if you are not diabetic. Your body should be able to handle that. If it's between 140 and 199 milligrams per deciliter after you have that glucose drink and we time it out for 2 hours, you are what's considered pre-diabetic. If it's above 199 milligrams per deciliter, you are what's considered overtly diabetic. So have them drink a glucose drink, and then we check their glucose values every 15 minutes and plot it on a line. In a clinic, they're just going to check it 2 hours later, but we don't mind actually making you prick your finger over and over again in order to do it, which is what one of you, as a volunteer for this lab, would have had to do. Now, when you think about a glucose tolerance test, what you're seeing here, notice that the fasting glucose at the very beginning in the diabetic subject is significantly higher. In fact, it exceeds the 125 milligrams per deciliter threshold. So that tells you right there that this person is overtly diabetic and this person is normoglycemic because it's between 70 and 100 at the beginning. They drink the glucose drink, right? They do that in 5 minutes. And then you look at what happens to their blood sugar. Here we get blood sugar spiking at about an hour. And then after about 2 hours, it's kind of still at that elevated spike. That's not good. What that indicates is that glucose is not moving into cells. Cells are not able to suck up glucose, right? If it's a type 2 diabetic, it means insulin isn't working and there are no GLUT4 transporters in the surface of the skeletal muscle cells or the adipose cells. If you're type 1 diabetic, it means there's no insulin and therefore there's no GLUT4 transporters in the surface of skeletal muscle cells and adipose cells. So that glucose has nowhere to go. Literally, it can't go anywhere. Cells can't use it. They can't actually use that glucose if there's no insulin. And here you see a quick spike and then this body brings it back down. What you're seeing here is skeletal muscle fibers and adipose cells with those GLUT4 transporters sucking up that glucose, and they suck it out of the cardiovascular system. So another pattern I want you to be aware of is what's called reactive hypoglycemia. There are some people who, after a glucose tolerance test or after they eat, will feel really good. They'll have kind of a high, and then they will crash. This is because they get an exaggerated insulin response that actually brings their blood glucose levels to hypoglycemic ranges, and it takes a little while for their body to recalibrate and bring them back up. This condition is called reactive hypoglycemia. So if you ever hear somebody go, "I'm hypoglycemic," this is a form of that. And because things like neurons need glucose to burn, right, it can cause you, if you're reactive enough, it can actually cause you to lose consciousness. So one of the things you're going to see over and over and over again if you continue on your nursing degree is you're going to see diagrams that look like this. And these are essentially pathology diagrams or pathophysiology diagrams. And they're a consolidated way to talk about a broad, huge subject. And this is a very good diagram, in my opinion. So we're going to focus on type 1 diabetes really quickly, but a lot of what we talk about applies to type 2 diabetes as well. And we're going to talk about how diabetes can lead to loss of life, because a lot of people die from diabetic complications. Now, we know glucose is sticky, and it just sticks to everything, which can cause all sorts of problems. But let's look at a few other things. So if you eat a meal and your plasma glucose levels go up but no insulin's released, that glucose is just going to continue floating around in the body, right? It's going to be in the blood and the interstitial fluid, but your cells aren't going to be able to use it. It's like being in an ocean of salt water, right? You can't drink any of it. It's all over the place. Water is all over the place, but you can't use any of it, right? Same thing here. Glucose is everywhere, but the cells can't physically get the glucose into the cell. So if glucose can't get into the cell, one of the things that the body does is it begins to break down fat. So it begins to actually physically break down fat. So if you remember that kid that looked very emaciated, right, despite eating like a horse, the reason why is they can't use glucose, so their body dips into their fat reserves. So you're going to see wasting. You're going to start to see wasting in type 1 diabetes, untreated. Another thing that happens is if you're breaking down and using fat for energy, you produce ketone bodies as a byproduct. Ketone bodies are very, very acidic, and they can lead to what's called diabetic ketoacidosis. Diabetic ketoacidosis, if it's severe enough, can cause two things to happen. One, it can cause proteins to denature or unfold, which can interfere with neural mechanisms and all sorts of different body processes. You can slip into a coma and you can die from the acidosis. Another thing that can happen is in acidosis, the body tries to compensate the drop in pH by sucking hydrogen ions into cells and pumping out potassium ions. If potassium ions or potassium concentrations get too high, that can essentially stop your heart, and that can lead to death. So you have death from diabetic ketoacidosis, right? Now, if you're hyperglycemic, one of the things that glucose will do is it will suck water into the renal tubule. And as it sucks water into the renal tubule, it causes something called osmotic diuresis. So sucks water into the renal tubule, and it produces, because water follows solute, water is going to follow that glucose out of the body, and you're going to start urinating more. So you're going to start urinating and urinating and urinating, right? So if you're urinating all your blood volume out, your blood volume is going to drop and your blood pressure is going to drop. And when blood pressure drops, that can lead to circulatory failure, right? You can also die, not just from circulatory failure, but from dehydration. So you can actually die from the dehydration caused. You can urinate yourself to death, and it sounds funny, but it's absolutely something that can happen. So another thing that happens is as fat begins to break down and we utilize all our fat reserves, the body's last choice is to use proteins. So if you need to break down proteins for energy, one, your tissue is going to start wasting away. Two, proteins give off a lot of nasty byproducts, notably things like ammonia, and There you're just going to get wasting, and that's going to contribute to the overall poor health. The point of this is um, diabetes affects everything. It is a multi-system disorder that impacts every other system in your body. So final learning objective is glucagon. So we've talked about insulin, diabetes. Now we're going to talk about the role of glucagon. So glucagon, stimulus for glucagon release, low levels of blood glucose, right? Receptor alpha pancreatic cell, control center alpha pancreatic cell, effector the hormone glucagon. Glucagon's primary target is the liver. Within the liver, when glucagon binds to receptors on its target cells in the liver, it triggers a pathway that leads to a few different changes. One of them is the breakdown of glycogen. So you break glycogen down and release glucose molecules, and those glucose molecules go into the system. The other is gluconeogenesis, where you start making glucose from non-glucose precursors, and those, right, exit out those GLUT2 receptors and enter into the bloodstream, or GLUT2 transporters and enter into the bloodstream, right? So you got a couple of different things that are completely opposite, because now you want to make sure that you have adequate levels of glucose. So when you think about glucagon, think about the breakdown of glycogen and the gluconeogenesis, which is the formation of glucose from non-glucose precursors. So when glucagon binds, activates a G protein coupled receptor pathway that activates enzymes that upregulate these guys. Now, one of the medications that's used to treat type 2 diabetes is what's called metformin, and metformin has a target on a particular transporter. One of the things that metformin does is it activates different mechanisms in the cell that ultimately improve insulin sensitivity, improve glucose transport, reduce fatty acid synthesis, so it reduces the amount of fat that's released, and it improves insulin sensitivity. This is the number one recommended drug for type 2 diabetics. If you pay attention to some of the literature, it's also one of the few medications that's ever been demonstrated to extend life. So a lot of people who work in the field of aging and life extension actually take metformin. They're not prescribed it. They don't have type 2 diabetes. But something about very low dose metformin can actually improve people's overall metabolic health. And there's a lot of interesting podcasts about it. All right. Now, another thing to realize is that glucagon can work synergistically with other hormones that elevate blood glucose. So remember that cortisol can trigger elevations in blood glucose, epinephrine. When all three of them are working together, you can get these big, huge spikes in blood glucose. And that's a way to ensure that if there is a situation in which your muscles and the different tissues in your body need glucose, if you activate your fight or flight systems, right? Working with this glucagon here, you ensure that the body has plenty of glucose reserves. All right, thanks. Bye.